Just now you have seen the photograph of start tank reactor and the plug flow reactor. The start tank reactor can be used both the mode, batch and continuous mode, and plug flow reactor mostly used in a continuous mode. Now this course deals with aspects of biochemical engineering, and as you know that purpose of engineering is a kind of application of science. As for example, that from physics, the courses like electrical engineering, electronics engineering that has been developed. Then if you look at that material sciences, on material sciences we have mechanical engineering, we have the metallurgical engineering, and from chemistry the engineering courses like chemical engineering that has been developed. Now, if you look at the biochemical engineering, it has some resemblance with the chemical engineering, and if you look at the chemical process and biochemical process, there is some differences. The chemical process, when we produce anything, that as your product changes, your substrate also changes. Now, in case of biochemical engineering, biochemical processes, we can produce different type of products from the same substrate. So, that is the specialty of this biochemical processes. Now, so basically initially this course will discuss that what is the stoichiometry of the bioprocesses, and after that we will discuss the kinetics of the chemical reaction kinetics, then chemical reactor analysis, and it will be when we have some information on the chemical processes, then we switch over to the biochemical process. And if you look at biochemical process, we have two type of kinetics that is involved. One is enzymatic reaction kinetics, another is microbial growth kinetics. Now, in case of enzymatic reaction kinetics, as you know, enzymes they are very specific with respect to substrate, and they give a particular products. I can give the example that glucose isomerase enzyme, when acts on glucose, is produce fructose. But when you go to talk about the microbial process, they are also they produce but this is usually formed with the help of metabolic pathways, and in the metabolic pathways we target a definite type of products, but with that products also some other metabolite formation is there. I can give the example of citric acid fermentation process, where the Aspergillus niger is used glucose for the production of citric acid. Now, when citric acid produced, there will be trace other metabolites formation there, but in case of enzymatic reaction kinetics, we do not have the byproducts, we have only one product, because substrate is very specific with respect to substrate. So, your product also will be that product formation will be there, other than no byproduct formation take place. Biochemical reaction usually carried out in the batch process as well as continuous process. Now, in continuous process, we have two type of reactor. We have continuous start tank reactor, and there is a plug flow reactor. Now, this batch process is usually carried out, it can be operated very easily. You take the material at a time, let it react, and after the reaction is over, you take out the product. But in the continuous process, as for example, when you work with CSTR, and CSTR when it works with the biochemical system, we call it chemostat. Now, in the chemostat, when we operate any chemostat, first we start operating in the batch mode. When your cell grow maximum, then we start feeding the substrate. Now, and then we operate it for infinite period of time to have the that steady state conditions. Now, I can give you some instances that that will be very clear. Here, if you look at this reactor, this is here we are working on algal fermentation process. Now, this is the start tank reactor, and this is the starter that we have, and purpose of the starter to make the homogeneity in the reaction mixture, and this can be operated both in the batch mode as well as in the continuous mode. And we have this is a control panel, and in the control panel we can control the agitated speed, we can control the temperature, we can control the pH. So, that we know the living system or biological system, they have special characteristics. As for example, they are very sensitive to the environment, they have the acclimatization property and they produce some kind of waste product. So, so after that, here there is the example of the plug flow reactor. In the plug flow reactor, there is no back mixing. You pass your substrate one end and take out the product in other end. And another type of start tank reactor here we have, where that we can this can be used both in the batch mode and continuous mode. Now, when you talk about the purpose of the engineering, the aspects of biochemical engineering, one is the important aspect, that is the scale up of bioprocess. Suppose whatever we develop in the lab scale, how we can do it in the commercial scale. Now, when you transfer this scale from the lab scale to the commercial scale, the operational parameter of this process will be changed, and how this operational parameter will be changed, that we will be discussing in details. Now, if you look at this fermenter, another very important aspect of the biochemical processes is the sterility of the process. Now, most of the biological process, they are aerobic fermentation. So, you have to spurge air. So, air should be sterilized, and media that we this media where we allow our desired organism to grow. So, sterility of the fermenter is to be maintained. So, this is the air and medium sterilization is a very important aspects of this particular coursework. And another thing, ultimate that our destination is that how industrially these processes are operated. So, this course will give you some idea that application of this particular biochemical process at the industrial level. And if you look at to this course, in overall it deals with several things. First, initially it will cover little bit of microbiology and biochemistry, then followed by the what are the different type of bioproducts is produced, the biological process, classification of bioproducts. Then we will give the difference between the chemical and the biochemical processes, how it differ from each other. Then we try to understand chemical reaction engineering, and then from that we will try to find out the chemical reactor analysis. Then we switch over to the biochemical process, where we will analyze the bioreactors, and these bioreactors with respect to pure enzymatic reaction, with respect to your microbial cells. After having this idea, also this course will give you some idea on the stoichiometry of the bioprocess. Then question comes that how different models we have in this biological system. As for example, we have monod growth model, and we have part model, we have Ludeking Pirate model for the product formation. How those model can be implemented to understand the process in a better way. And finally, we will do the scale up of the process. We try to find out how we can scale this process from the lab scale to the commercial scale. So, this course will be very much useful, not only for the biotechnology student, but also used for the biochemical engineering student, chemical engineering student, environmental biotechnology student, biochemistry student, also a microbiology student.